Why is the Human Genome Project an important milestone in the study of biology? What is the Human Genome Project? The Human Genome Project is an international $3 billion scientific research project which aims to identify and sequence all the genes of the human genome. A genome is the complete DNA sequence of an organism, including all of its genes. It is the largest collaborative biological project undertaken to date. The Human Genome Project began in 1990 with the aim of sequencing the DNA of the human genome in 15 years. In 1998, a private company, Celera Genomics, was established and embarked on a parallel project. This led to competition between the Human Genome Project and Celera, which increased pressure on the project to be the first to sequence the human genome. By 1999, chromosome 22 became the first human chromosome to be sequenced. In 2001, the first draft of the human genome was published, and in April 2003, 13 years after the project began, a more complete draft was published. The project was declared complete. What techniques were used by the Human Genome Project? The Human Genome Project made use of and indeed developed several DNA sequencing techniques, which made the project possible. The main techniques used are map-based sequencing, whole genome shotgun sequencing, the Sanger method, and recombinant DNA. Map-based sequencing, or BAC to BAC sequencing, was the first method of whole genome sequencing developed. Using this method, a crude map of the whole genome is created before sequencing can occur. Human DNA is cut into large fragments using restriction enzymes. These fragments overlap slightly at each end. These fragments are cloned in bacteria, vectors, which store and replicate the human DNA. These clones are known as bacterial artificial chromosomes, or BAC clones. This is so that enough clones can be prepared so sequencing the order of the clones can occur. The overlaps in these fragments were used to order each fragment's place in the genome, creating a map of the entire genome. In order to sequence the DNA of these clones, they are cut into even smaller fragments called subclones. A sequencing reaction is carried out on these subclones, which are then put into a sequencer. A computer assembles these short sequences of base pairs into a contiguous sequences, representing the human DNA in the BAC clone. Put all these sequences of subclones and BAC clones together, and you have sequenced the whole genome. Although the map-based method provides very accurate results, the necessity of mapping the genome is very time-consuming. Advances in sequencing technology and software enabled the use of the whole genome shotgun sequencing method. This method bypasses the mapping stages, making it much faster, although criticized for being foolhardy by some. The whole genome was cut into small overlapping fragments which were directly sequenced. Then the whole genome was put together using just the overlaps of these fragments to order them. The Sanger method is used by both of these genome sequencing methodology. A polymerase chain reaction produces millions of cloned fragments of DNA. Then, substances are added randomly to stop the creation of DNA at each of the 4 bases. The result is strands of DNA of almost every possible length. The length of these strands are determined by making use of gel electrophoresis. Markers on each of these strands shows which base is at the end of each strand. When the result is combined, the sequence of bases at any point can be found. This process was highly automated during the project and was very efficient at sequencing the bases of the small fragments of DNA used to map the genome. What is the significance of the Human Genome Project? The completion of the Human Genome Project has led to benefits in many different fields. In the medical field, sequencing of the human genome has allowed for greater understanding of and treatment of diseases. The genetic makeup of an individual can increase susceptibility to certain diseases. DNA-based diagnostics makes it possible to detect mutant genes which predispose to these diseases. Therefore, early intervention or use of personalized medication and gene therapy makes it possible to limit the impact of diseases such as cystic fibrosis. Benefits to the medical industry which the Human Genome Project is responsible for include improved design of medications and prediction of their effects, genotyping specific viruses to direct adequate treatment, and identifying mutations related to different forms of cancer. The Human Genome Project has dramatically fueled the growth of the industry of genomics, an industry now valued at $800 million, employing 4 million people. In forensics, technology developed by the project has led to DNA fingerprinting. This has been used in crime investigation to catch criminals, paternity and human ancestry and evolution studies. However, with these benefits, ethical concerns should be considered. Misuse of genetic information may lead to violation of genetic privacy. For example, the genetic information of an individual may influence the insurance policies deemed to them by an insurance company. Limitations of the data gathered. The data gathered by the Human Genome Project is in the form of base sequences. These sequences on their own are meaningless and require interpretation. Only a small portion of the base sequence of a chromosome actually consists of genes that are expressed. Genes can be identified by searching the sequences of base pairs. However, the proteins they code for and under what conditions the gene is active cannot be determined from the data alone. Rather, the function of the polypeptide the gene codes for has to be found in the human body. Many genes have multiple effects or interact in complex ways with other genes. Accordingly, many diseases are not caused by a single gene, so even though we have the base sequences, we do not know which genes or how many genes are collectively responsible for complex multiple gene diseases such as diabetes or bipolar disorder. Another limitation of the project is that opposed to the initial hope of accelerating the development of new treatments to diseases, it requires more than just the knowledge of base sequences to cure diseases. Why genetic linkage maps could not be used to map the human genome? Genetic linkage refers to the tendency of alleles that are close to each other on a chromosome to be inherited together during meiosis. The closer two genes are on the same chromosome, the higher the chance that they will be inherited together. These maps are constructed by observing the frequency of crossing over between two genes. A genetic linkage map shows the position of known genes relative to each other in relation to recombination frequency. The Human Genome Project could not have been undertaken using genetic linkage maps alone for the following reasons. First of all, it is impossible to make controlled crosses in humans without crossing a ridiculous amount of ethical, legal, and moral values. Therefore, geneticists have to calculate recombinant frequencies by chance dihybrids in human matings. Also, the human genome is immense, meaning average distances are very large. Obtaining enough data to map the entire human genome using this method would be an exceptionally difficult task. Furthermore, as they do not give the exact location of genes on a chromosome or the DNA-based sequence, linkage maps alone would not be able to achieve the primary objectives of the project. In conclusion, the Human Genome Project was highly successful and, despite its limitations, has opened the door for a myriad of genetic research and biotechnologies, which may solve problems facing humanity in the future.